So thank you, Olivier, and the organizers for inviting us and for making this meeting possible. It's great to see you again, guys, after 2 years behind a computer screen. So we will talk about a simple model of neural network with short plasticity. I will present the easy part, and then Eva will present the serious part. So I will talk about delayed responses. I will tell you what it is and the link it has with, or we think it has with, working memory and persistent activity. And then Eva will go with the actual maths later on. That's a joint work with Antonio Gales from Sao Paulo and Enrico Presciuti from L'Aquila. And we start the story with Joachim Förster in 1973, who wrote in the first paper that described the phenomenon I will show you next. A delayed response trial typically consists of a presentation of one of two possible visual cues, an ensuing period of enforced delay, and at the end of it, a choice of motor response in accord with cue. The temporal separation between cue and response is a principal element, making the delayed response procedure a test of an operationally defined short-term memory function. So what does that mean? You have a scheme here of a monkey. At the beginning of the experiment, in this region here, the monkey can see the content of two boxes. Okay, that's a glass window. The two boxes are opened. One of them typically will have a reward. And that's the cue period that lasts a few seconds. Then the blind is lowered. The monkey cannot put its hand at that time around the boxes, okay? And for variable delay, it has to wait and to remember in which of the two boxes the reward was. After a delay, in that case it's a bit more than 30 seconds, the blind is removed, and then the monkey is allowed to put its hand in this region and to open one of the boxes. And if it gets the right box, it has the right to get the reward. And after conditioning the monkey, Förster started to record from the prefrontal cortex, and the ticks that you see here are spikes from a given neuron. Okay, these are several trials, and you see that the neuron that is recorded right now, that's so at the same neuron for 5 successive trials, is weakly active spontaneously. When the cue comes, the activity doesn't change much, but during the delayed period, so while the monkey has to remember where the reward was, the activity of the neuron increases a lot. And when the blind is left, the activity disappears. What CR and CL here mean is that the reward is in the right box or in the left box. So you see that for this neuron, it doesn't matter in which box the reward is, just that there's a reward to remember. More detailed picture with some trials this time where there's nothing in the boxes. Okay, there's no reward at all, and you see that in that case, you don't see the sustained activity of a neuron. Okay, it's only when there's a specific box to remember that the persistent activity appears. So that was done in 1973. And so it's not only to pay homage to this great experimentalist, but I show you this old data. It's just to make clear that this has been studied for a long time. And if it has been studied for a long time, it's because it's easy to reproduce. Okay, So never take at face value what a neurophysiologist tells you. He or she has a need to publish his papers and to get his stories accepted. So they tend to oversell what they do. This has been studied for 50 years now, so we know that it's robust. And it has been studied by many different labs, as you can see here. And that means it's not too hard to find these neurons. And if it's not too hard to find these neurons, given that you lower down the electrode in the prefrontal cortex, you don't know exactly where you bring the electrode down. That means there's many neurons that exhibit this kind of activity. So that's the modern paradigm that is used. Now people train monkeys to fixate a spot in the center of the computer screen. The cue becomes one target on a circle, so discrete targets. During the delay period, the monkey must keep its eyes on the center, and when the signal comes, bell rings typically, the monkey must make a saccade towards where the cue was, and then it can get some orange juice. And when you do this more sophisticated version of the experiment, you see this kind of responses. So again, each tick marks the time of a spike of a single neuron. That's the same neuron that you see on all these trials. And you see that you observed sustained activity only when the cue is in the south, okay? And you have a slightly decreased activity when it's in the north. So in that, it's more sophisticated, and here we have the impression that we see a neuron that is specific for the target location. The same, larger, and to make one important point clear, you can change the delay, and the sustained activity remains for the entire delay period. Okay? What happens if it happens, when the delayed sustained activity disappears? That's shown here in two examples. So the first three trials, the cue is on the west side, the activity persists, the signal comes, and the monkey does the proper saccade. In this trial, you see that the persistent activity spontaneously disappears. And when the signal comes, the monkey makes a wrong saccade. Other example. Here, the persistent activity is there, the target is northwest, and the monkey does a proper saccade. In that case, the activity doesn't appear at the right time, and when the signal comes, the monkey moves the eyes to the south. So there seems to be a link between this sustained activity and the fact that the monkey remembers properly where the target is. And that's what we want to if we want to construct a model that can reproduce this kind of sustained activity with the simple dynamics for the neurons and simple connection patterns. People have kept working, as I said, on this kind of experiments. And here you have a more sophisticated kind where the monkey presses its finger, one of its fingers, on a plastic lever that starts vibrating. So the cue is vibration of a lever for 500 milliseconds. Then there's a delay period, and then vibration starts again, and the monkey must remember the frequency of a vibration. And what you see when you find one neuron that exhibits sustained activity during the delayed period is that now for these neurons, the rate of during the persistent activity is linked to the frequency of vibration. So people working on this specific type of working memory persistent activity call that parametric working memory. And that's not something we will deal with in our model. But that's something, so the phenomenology is fairly rich. And that's an experimental problem that has been one of the driving questions for the development of theoretical neuroscience. So as soon as people started working with more dynamical models than the Hopfield network, so people like Daniel Amit and Misha Tsolix, one of the first questions that they started to clarify was this persistent activity. And so you have an early, from 2001, review by Xiaojin Wang on the subject using linearized integrated fire neurons, networks of linearized integrated fire neurons. And he accounts for this persistent activity as a fixed point in a system of ordinary differential equations. So to make clear that we are not the first, far from that, to try to build models for this kind of question. So I showed you the behavioral data. Now, what about the neurons that are supposed to sustain this behavior? And you have a very interesting paper by another Wang and Mark Ram's group, and Patricia Goldman-Rakek was one of the key researchers working on this subject. So what they did, they used brain slices of prefrontal cortex from a ferret. They performed multi-neuron patch clamp recording. So these are very tough experiments. Okay, already getting a single recording working is a tough job. They have 6 electrodes at the same time. You need to have a good experimental setup and to be trained to do that properly. And what do they find? They find a subnetwork that contains complex pyramidal cells that displayed non-accommodating discharge. I will show you what it is. They were highly reciprocally connected, and their synapses were facilitating. So that's one of the neuronal pairs that we recorded. So you have a green pyramidal cell and a black one. You see, now you're all experts after the talks of yesterday of morphologies of pyramidal cells. You see that they have apical dendrites that branch fairly early. So you have essentially 2 apical dendrites each. The red dots are the potential synaptic connections between the black guy and the green guy. What you see on the right, so the tiny traces here are the spikes from the black guy. Be careful, the 2 cells are very different. Okay, so that's 200 millivolts, that's 0.5 millivolts. And at the top, you have a postsynaptic response in the green cell. And when you go from the top to the bottom, what happens is that they increase the rate at which the presynaptic neuron, the black guy, is excited. And you see that when you're excited at a high rate, the second response, even though it stays at the top of the first one, is larger than the first. And if you stop stimulating, wait for a while, stimulate again, you see that you have this response, which is much larger than that one. So you have facilitating synapses, which is not the typical phenology that we see between pyramidal cells. So these cells are very specific, they are highly interconnected, and their synapses exhibit short-term facilitation. And again, modelers have been using this information for a while. That's a review by Barak and Sodix. You can find on my website both the slides and the handout with all the references. You just have to click to get the papers, okay? So in what way does what we propose changes? We will use stochastic units. Why? Because as soon as you start looking at how neurons actually behave, everything is stochastic. So that's an example from a key paper from Fred Ziegworth and Henri in 1880. What you have, I've lost. No, it's here. Yeah, it's here. So what you see here are single traces, single trials. They brought a patch, so a glass capillary, a patch pipette, onto a cell. In that case, it's a muscle cell, in fact. And what they see is what goes on below this, within this small patch of membrane. And they depolarize the patch with this electrode. And they record the current they have to inject to keep the voltage at the preset level. At the top, nothing happens, and when it goes down, that means something happens, and this something is the opening of a single ion channel, a sodium channel. And these are successive trials, and you see sometimes the opening of the channel comes early, sometimes it comes late, sometimes it's very brief, sometimes it's longer. Okay, so what you're seeing here is a single macromolecule that is switching back and forth between two or more metastable states. Okay, and you can see it because when it's in the open state, you have a relatively large current flowing through the channel. If you make that 300 times and look at the average response, you get basically what Huxley saw. So that's the population response. Okay, and these channels are responsible for the upstroke of the action potential. So that means you expect to see some jittering, so fluctuations in the time at which the action potential occurs, even if you repeat many times the same stimulation. If you look now at the synaptic transmission, that's an easy preparation where the neuron makes a synapse onto itself. That's an OTAPS. So with a single pipette, you can record both the pre and the post-synaptic side because it's the same. And so concentrate on the B panel. That shows you successive responses to the same stimulation at a low rate. Sometimes nothing happens. Sometimes you have a small response. Sometimes you have a large one. And always at the top of these large fluctuations, you have tiny ones. So the tiny fluctuations come from the fact that ligand-gated channels at the synapse behave in the same way as a voltage-gated channel for the action potential. They fluctuate between an open and a closed state. And the large fluctuations come from the fact that sometimes you have no release at all. Sometimes you have a single vesicle. Sometimes you have more than a single vesicle. So and you see, and when you do experiments, that's very striking. And what convinced me to do neurophysiology was seeing this kind of behavior. I thought it was magical. So that's to make the point clear. We want to use stochastic units because every step you look at in the way neuron works is stochastic. And now to come back to this short-term plasticity that Wang and Markram showed within this population of pyramidal cells. So it acts on a time scale of the order of 100 to 1,000 milliseconds, so comparable to the spiking activity. And there's several mechanisms. We talk about short-term potentiation, but now you are all experts about these mechanisms. It's essentially when you look at chemical synapses, what provokes fusion of vesicles and so release of transmitter is entry of calcium in the presynaptic terminal. Once the calcium goes in, it has to be pumped out. It takes a little bit of time. If a new action potential arrives before all the previous calcium was removed, you have a slightly increased calcium concentration. And since the release probability depends on the fourth power of the calcium concentration, even a tiny residual calcium can give you a greatly enhanced release. And that's what is stated here. So before I give a microphone to Eva, I will just summarize what we want to do. We have this basic phenomenology. So the neuron is not reactive, and then there's a cue, and then we have high frequency activity that disappears when the signal comes. we have a network with many neurons. And within this network, we can find a subpopulation of neurons that are highly interconnected, and the synapses between these neurons exhibit short-term facilitation. Okay, so the model we built is restricted to this subpopulation of neurons that are highly interconnected. And because we saw that most of the building parts of the neural activity are stochastic, we use stochastic units.
Thank you, Christophe. So I follow on this with speaking more about the mathematical model that we have proposed. I will do it here because Etienne told me that I have to show things on the screen such that the people following on Zoom or on YouTube can also see. So together with Christophe, Antonio, and Enrico, we proposed a very simple mathematical stochastic model for short-term potentiation, which is due to the residual calcium, and in which we can describe the short-term memory of the system as a tendency of it to keep track of an initial stimulus. And this keeping track will be, in mathematical terms, actually the fact of, I'm not able to show it here. You think this is, they see it? Yeah. By the fact that the system stays in the vicinity of a certain untypical region of the state space, which corresponds actually to an attracting fixed point of some limit system. And because the system is finite, it will be finally kicked out of this region and will relax to its true stable state, which is not the same as this, what we will call a metastable state. Okay. The model is a simple model of spiking neurons, which are spiking according to a random point process. So N is the number of neurons. So there's a subsystem here. They are all interconnected. I represent each neuron by its membrane potential. We are in the purely excitatory case, so I just shifted all these membrane potentials to be positive. So UTN of I is the membrane potential of non-I at time t. And then spiking occurs according to a point process with a spiking rate that depends on the membrane potential through a non-linearity. So this little phi here is the rate function, so something like Acus tangent. And that just means that the probability of spiking within a small time interval after time t of length h, given the whole history, that means also all the interactions, is just the function phi of the current membrane potential times the length of the interval. Okay. So in other words, these are exponentially distributed waiting times for the next spikes, but the parameter of the exponential distribution is random since it's evolving over time. So it's not a true parameter. So it's this phi of ut. Okay. So we call it the spiking rate function. Yesterday in Quant's talk, there were Hawkes processes. Actually, this will also be Hawkes processes, kind of Hawkes processes. So in his case, the phi function was just constant plus x, okay? And here it could be something more complicated. What is important, we suppose that phi of 0 is 0. That means if there is no input, there is no spiking, okay? So this will in particular imply that 0, so the silent state, is an equilibrium state. Okay, so it's an isolated system. We suppose it's exposed to some stimulus in the beginning, but then we stop the stimulus and wait in the long run what is happening. Okay. So, good. I write zt for the number of spikes of neuron I up to time t. And each time I spikes, it gives an additional amount of stimulus to all the others. And we call this SP for wi to j. From the others, we see this additional amount of potential. And this W depends actually on time, and it depends on the residual calcium concentration right at the time of the neuron itself. So more precisely, we will take it as a function alpha just a constant over n, number of particles, times the residual concentration of neuron I at that time. Okay, so the synaptic rates are modulated by the current calcium concentration of the synapse, of the given neuron, which is I. Okay? And then in between successive spikes, there is a continuous leakage of the membrane potential as usual, such that you get an equation of this type. So the membrane potential of a given neuron in between successive spikes is decreasing exponentially fast. And then you get kicks, which are of order alpha over n, but more modulated by the residual calcium concentration of the presynaptic neuron J. Okay, and then the exponential loss of potential also. Okay, and then for the calcium concentration, we also just impose a simple point process. As Christophe said, it's acting on the same time scale as the spiking activity. So we just increase the residual concentration by 1 at each spike of the neuron, and then we have some leakage also in between successive spikes. Okay, so we have this very simple equation here. So the residual concentration of neuron I decreases and gets a kick of height 1 or whatever you want, something macroscopic, each time the neuron I is spiking itself. Because of the fact that we suppose that the spike makes that you have this calcium concentration which is remaining. Okay. Good. Any questions so far? So very easy model. Just a comment for those who know preceding works we have been doing with Antonio. Usually we look at models which we call being of variable memory, which means a neuron which is spiking, we reset it to 0 or to resting potential. We don't do this here, it's for mathematical reasons. That's the only, okay, because we want to have a limit system which has a deterministic dynamical system behind such that we can write things down easily. That's the only reason. So no reset. And another thing, so this is the purely excitatory case. That's not really important. It's somehow motivated by Christophe's introduction. Okay, so we are not, it would be no problem to add inhibitory.
May I say something of hello, Eva? I'm Antonio Gobbs.
No, it's like a voice coming from
yes, from very far away. But this does not mean we don't believe the result would hold also for the process with variable length memory.
Oh yeah, of course, but you are impatient. I will say that, but a little bit later. So we believe that everything I will tell you is also true for the model with reset, but we did not yet prove it. But we believe it's true. There's no reason not to believe it. Yeah. Thank you, Antonio. Okay, for the people working on hogs, so you can see this as a system of n interacting pairs of Hawkes processes. So the counting process for each neuron would be the Hawkes process. And then, of course, you have this dependency on the modulation factor, which is the residual calcium, but which is just the functional of the underlying Hawkes. Okay, so it's a sort of 2n-dimensional Hawkes process. And so it's easy to study. So for the finite neuron system, as I said, since I said it's an isolated system, if there is no input, the firing rate is 0. And that also actually implies that the all-zero state is the only invariant state of the system. It's not very interesting, but I said we suppose we have this isolated system and we just ask ourselves, how long does it take time that an initial stimulus survives in the system somehow? Okay. And I think the first time that this was rigorously proven is in an article by Arlini and Guilherme, 2016, who proved that the only mathematical condition you need to impose is phi of 0 is 0, and phi is differentiable in 0. Then if you have this exponential loss, this immediately implies that almost surely the system has a last spiking time, which is finite. Okay, and after that last spike, it never spikes again and just just decays to zero. Okay, I don't give you the proof. It's beautiful, but there's no time for that. But of course, the situation changes as n tends to infinity. So if I go to an infinite limit system, actually, we will see the appearance of other long-time behaviors, and you see that immediately on simulations. So this is a simulation, of course, of a finite system. Christophe has simulated only 1,000 neurons. So here's time evolving, and this is about both of the residual calcium and the membrane potential. And you see, I don't even find the trajectory which is dying. I think it's somewhere here. Yeah. But most of the trajectories seem to be attracted to this part of the state space, which clearly is not the silent activity, and which we would say is something like a metastable state or quasi-stationary distribution, whatever. So like a stable state for a long while, okay, which would express the tendency of the system to keep track of the initial stimulus. And of course, we see this, we can prove this mathematically by passing to a large population limit and by identifying this point here as a second fixed point, as usual. So this is what I'm going to do now. I apologize for the slides. I wanted to be precise. So if you want to pass to the limit, it is convenient to write the evolution of the membrane potential processes by representing them written by some Poisson noise, okay? So I said I have exponential rating times. If you suppose that, for example, the phi, the rate functions is bounded, Quan has told you that you can simulate everything by using Ogata type of algorithm. That just means that you have to take a Poisson process, independent ones for each nuance, and then you can simulate them like this. This is what is basically written here. So these capital M's are the Poisson noises. I have a second component. So dt is just the times of arrivals. And the Z component is the acceptance or rejection variable that you need because your weights are not fixed. They are evolving over time. And that's why you have this indicator here, Z, smaller than the actual potential value seen through the spiking rate function. Okay. So why this is convenient? Because it will be a nice tool to write down the finite neuron system together with the limit system using the same noise. This is an idea which has appeared in the lectures on propagation of chaos of Snitman, I guess. He used that with Brownian motion, but it's the same idea for Poisson noise. Okay. So this is written here. So the limit as n tends to infinity should be obtained by something that people in physics call propagation of chaos. Propagation of chaos means if you suppose that initially you have chaotic conditions. Chaoticity means that all the neurons have IID conditions. That's the most chaotic situation you can imagine. Then this independence, the chaoticity, is transported as time evolves by the dynamic as in the limit. Okay. In other words, that means that you can approximate. You see, you have this interaction term here. So the ith neuron has an input which is given by some empirical mean here, which is the empirical mean over time of the calcium residuals of the others. Okay, so the idea is just since the interactions are of order 1 over n, this will be the same thing like an expectation in the limit, and that's why I wrote it here. So this empirical mean in the limit will be something like a theoretical mean, which means an expectation with respect to the typical behavior of a single neuron in the limit equation. And this expectation should then be this factor alpha times the mean spiking rate of any neuron in the limit system modulated by the residual calcium. Okay, so that's why you have an expectation of the spiking rate times the modulation factor, and the product of the two of them is inside the expectation. This is important because it gives rise to a non-Lipschitz contribution. Okay, so in the limit system, any neuron should behave as a copy, as an independent copy of this system of equations. So still U and R. If you have a look on it, U is a completely deterministic equation because we have the exponential loss, and then the gain, which is due to the mean gain of the total system. And of course, the residual calcium stays stochastic because there's no reason why that should become deterministic. Okay, good. So this is our limit model. I will write it again on the next slide. So the equation for R is very easy. Okay. So the only thing we have to care about is this factor here, because it gives rise to a non-Lipschitz contribution. And usually, you know, if you have to show by pk iteration or whatever that some differential equation has a solution, it's nice to have Lipschitz terms. It's always easier. Good. So here's the limit system once more. 2 remarks. So for the membrane potential of a typical neuron, the only randomness remains in its initial position. So we could say that's mathematical. It's a mathematical question. Do we have to impose this initial condition to be deterministic or stochastic, whatever? Because if it would be deterministic, then this is all deterministic, which means that in the limit, a typical neuron has a spike appearance process, which is an inhomogeneous Poisson process, okay, in this case. Okay. So in the sequel, I will speak about 3 questions. So the first question is conditions for the well-posedness of the limit process. Then how does the finite system converge to the limit? And then the question, how is the long-time behavior of the finite system related to the long-time behavior of the limit system? So I start with the first one. So as I said, we have to take care of this non-Lipschitz term, and I wanted to show you 2 kinds of answers. So first answer is easy. As I said, if I suppose that in the limit system the initial condition is deterministic, then U is a deterministic quantity. And in this case, this expectation here, I can take the phi of U out of the expectation because it's deterministic. So I get phi of UD times the expectation of R. And if you have a closer look to this equation for the residual calcium, you see immediately that you can take expectation and that you get something which is a closed formula for the expectation of R. Okay. So in this case, I get a very nice formula for little ut, which is now expectation of ut, and little rt, which is the expectation of rt. This is of this form. I still have the product term of phi of u and of r. But if I suppose that phi is something like not sublinear, but sub square root, I don't know if this is the right expression, certainly not. So phi of x is increasing at most as square root of x, then a simple change of variable formula. You replace u by square root of ut for large values of u, shows you that you have unique existence of the system and non-explosion. Actually, you first show that you have non-explosion, and then you have Lipschitz continuity on compacts, which implies unique existence. Okay. So this is somehow under the strong assumption that U0 is deterministic. If it's not, we still can do things just to show you. So if U is random initially, then it stays random because of this initial randomness. But if we suppose that we have exponential moments for the initial calcium, and if phi is bounded, which is not completely unnatural, well, then you can use the usual truncation argument. You see, if I compare 2 solutions, U R and U tilde R tilde, and their non-Lipschitz contributions, if I truncate the large values of R, by little r here, which is a fixed truncation value, so you get the contribution of R where it overshoots this truncation value, plus the existence of exponential moments for RT uniformly over time, then actually we get a control of the difference of the solution in expectation in L1 norm, which is of the order x log x. There is a tilde here, okay? And then there is a famous x log x extension of the Grunwald lemma, which is called Osgood's lemma, which helps you to deduce actually the unique existence. And you use the same kind of arguments to show also the convergence to the limit system in this frame. So this is on this slide. It's awful, but just to tell you that. So if I take now the finite neuron system and I couple it with the limit system, so I use the same Poisson random measures for the finite system of neurons and for the infinite ones, I can couple them together under these conditions on initial exponential moments for the residual calcium values, and I get a very bad weight in n, which actually I'm not able to control. Okay, but still we know it converges over finite time intervals. We can improve that a little bit if we suppose a little bit more than exponential moments initially for R0. If there's a little bit more, like this, then I can improve that to something which is almost the best weight, which is almost 1 over square root of n. Okay. Good. This relies actually on the fact that in this case, we can show that this function here is a Lyapunov function for the residual calcium, such that we have moments uniformly over time for the residual calcium. Okay. But actually, this is maybe too complicated. So in all the simulations that I will show you in the following, we finally suppose that the initial values of the potentials are all deterministic. Okay. And if we moreover suppose that they are all the same, since the neurons which are spiking are not reset to 0, finally we can all replace them by one typical particle, which is then evolving according to this equation. And you see that the only thing that we have to care of is once more this empirical mean. And so we need a control on the deviations of this empirical mean from the limit expectation. And to do so, we use Bernstein deviation inequalities for discontinuous martingales. And this is actually a kind of classical tool which really applies. So I don't write here the exact formulation, but if we impose that initially we have exponential moments, then both the membrane potential processes and these empirical means of the residual calcium concentrations stay within finite time intervals in small tubes around their limit quantities, the ones of the limit process that I have discussed. And this happens with exponentially high probability, which means the probability that this is not true would be smaller than exponential minus n to some power times a constant. Okay. And the tube that I put around my limit trajectory is, as you would expect, of order 1 over square root of n, a little less. Okay, so I lose a little bit. Okay. So I have deviation inequalities which are uniform over time intervals. And so the question now is really, how does the limit system behave in the long run? So I just recalled it here. So in the case where I have initially deterministic initial conditions, it's a simple two-dimensional dynamical system. You can immediately solve it for and try to see what fixed points you have. So any fixed point must satisfy these equations here. Of course, 0, 0 is still a solution, and any other solution has to satisfy this equation for you. Okay. So I tried to make a plot here of the null lines. So this guy here is the U null line. This is the R null line, and also I plotted the phase portrait, I guess one says in English. And you see that actually, so this is the 0 point, which is of course attracting. It is attracting. There is a second one which is not attracting, and then the third fixed point, which we will call Umax, Rmax, which is an attracting point. So, okay. And then, and it's actually for this picture that we decided to work with the Hawkes process, because otherwise we wouldn't have this explicit limit system. Okay. So if we expose our system to some initial stimulus that puts it into this region of the state space, then certainly it will stay there for some time, because as I told you, it stays in tubes around the limit system over any fixed time interval. Okay. This is a simulation of the phase portrait of the limit process. And this is the theorem which tells you once more, so the deviations. So if I put some initial conditions in the domain of attraction of this second attracting fixed point, and if I take T1 the first time that my limit system is in a small ball around the limit attracting point, then the probability that at the same time the finite system is not in this ball, or the empirical mean of the calcium values is not in this ball, is exponentially small. Or in other words, the finite system is imitating the limit system with an exponentially high probability. Okay. And this is what we interpret as an expression of this short-term memory. So I already said it. So I suppose that initially we have some stimulus which pushes the system into this domain of attraction. We switch the stimulus off and we start observing the system. But since the point is attracting, the limit system is attracted to a point, and so the finite system follows the limit system at least over finite time intervals. And this behavior we interpret it as a short-term memory. Of course, in the long run, the system will tend to the zero state, but this is not what we saw in the simulations. At least not in the first one that I showed you. Okay. So here are simulations of the system. Christophe simulated 1,000 neurons. Okay, so on the left-hand side, you have really the stochastic system. On the right-hand side, you have the limit system. You see it looking nicely, fairly the same. The next slide is about the question of Antonio. So of course, if I add the reset of each variable, I wouldn't be able to say what is the limit system exactly behaving like, because I don't have a dynamical system. But what Christophe did is he just plotted here the same phase portrait of the Hawkes limit system, but the simulations here, you see these figures here, this is the system with reset. So Hawkes processes, but each neuron which spikes is going back to zero. So it's not attracted to the same point, of course, because there must be some loss, but it looks as if it's clearly attracted to another attracting fixed point, which is not zero. Okay. So we believe that that it's also possible to prove this mathematically, but we did not do that yet. Okay. And of course, this should somehow be related to metastability. I have already mentioned that notion. I like to quote this. I think the first time that I saw this quote is in a talk by Morgan. Possible, no? So metastability has maybe first been defined in a rigorous way by two physicists, Penrose and Leibowitz. And it's three things that it should express. First of all, if you start your system close to a metastable state, because it's metastable, it should remain there for a very long time. Okay. However, once it gets out of the vicinity, so of a ball around this metastable state, it should not go back there. So the probability of going back is small. And the exit time of the neighborhood of the metastable state is unpredictable. And unpredictable means in probabilistic terms that it's an exponentially distributed random variable. Okay. which you can't predict. If you renormalize by the expectation of the total length. So in other words, if I write tau n, because it depends on the number of particles, for the time that my system needs to leave this small ball, or maybe a bigger ball around this equilibrium point, the U max, then we should have convergence in law of this tau and divided by its expectation to some exponentially distributed random variable. Okay, and we believe that this is true in our model, but once more, or a similar result for the last spiking time, because I showed you the result by Alini and Guilherme. There is a last spiking time, so last spiking time divided by its expectation. That also should converge to some exponential one distributed random variable. We see this still on our simulations. So this is, of course, we can't really prove it because n is finite. So what Christophe did is just he plotted the survival function, the empirical survival function of the system, which is the probability of having survived up to some time. And it's not probability, of course, it's the empirical probability, which means it's just the fraction of neurons that are still alive. So how do we decide that neurons are alive? Just by saying that their spiking rate is above some threshold, which you chose to be equal to what? No, actually. Yeah. Okay. Good. So we believe this is true. Actually, we proved that in a different model with Pierre Montmarché, this metastability result. So it's possible to prove it, but it's a lot of work. I already mentioned the deviation inequalities we used for the proofs, but maybe I skipped them. And I just close with some literature. I wanted to acknowledge the nice article by Alini and Guilherme. And then 2 articles about the short-term synaptic plasticity. And if you want to read our joint work, it's on arXiv. It has also appeared in Journal of Statistical Physics. Thank you. Attention.
Thank you for these very interesting presentations, both of them. We have a question from the YouTube channel, but any question from the room? No? Yes?
Thank you very much for this very nice talk. Maybe my question is naive, I would say. But with respect to this metastability result that you were mentioned at the end, do you believe that there is a relation between phi and alpha, the leakage rate? Because I was wondering, well, my guess is,
anyhow, it's
if we imagine the spiking probability when the memory potential is very small, it could be like approximately phi of 0 times u somehow. And then if alpha, the leakage rate, is much smaller than this, it should be more difficult to somehow die or stop spiking than the opposite. Would such a condition be necessary or not? Because if you understood, during the simulations you always die and...
Well, in the simulation, anyhow, you always die. But your question, of course, if alpha is too big, then the second fixed point will not exist.
Mm-hmm.
Okay.
Okay.
So we need alpha to be sufficiently small.
Also, to observe the system dying in the simulation, I mean, for the survival study, the network size is 20. Okay, I mean, if we take a network with 1,000 or 10,000 neurons close to the fixed point, we don't observe the system escaping.
Mm-hmm. Maybe may I ask another question? Okay, no. Yeah, what about could you introduce some spatial structure in the model as well? Would the analysis be similar or yeah? Okay.
Oh, sorry. You said yes? Yes. Okay. Etienne?
Okay, thank you. Do you have any idea of the order of E of tau n as the dependence in n? Short answer, no.
It would be exponential. I guess it's exponential. Yeah, but so these are simulations I run on my laptop now. So I have to try. I can.
Thank you. Well, maybe person from the YouTube channel, Romain, are you here?
Yes. Hello. Thank you for this very combined talk. A nice talk. I have one technical question. I did not understand why the nerve lines do not intersect at 0. They should know if 0 is an equilibrium.
Yeah, it's huh?
Maybe, yeah. No?
Yes, they should. I think it depends on there's a discontinuity of, you see what I wrote here? I mean, the U-null line is only for U strictly positive, and it's defined by this capital phi. I don't remember the calculus, but it's sure it's true, which is U over phi of U. You see? So,
yes.
so there is a discontinuity. So maybe my drawing is something wrong. Yeah, it does intersect because I wrote here R equal U equal to 0.
Okay, so maybe you can discuss this offline.
It's a technical question.
And
I guess
I have a better question. So I was wondering, how far are you from the 2D Markram model for short-term plasticity? You seem to have designed your own, basically. So
to the Markram.
So when you speak about the Markram model, what do you mean exactly? So something that fits the developmental data I showed, or something?
Yeah, they have some. Michal Chodix have written several papers on this data with a specific model, and it has synaptic depression, potentiation. It's basically a PDMP where you have depletion of vesicles, but you have also, addition of vesicle to a pool. So it sounds like it's a bit similar to what you do, but
yeah. So what we do is much simpler. We have only facilitation, and we could add an extra variable, so extra to the R that would model depletion. That would not be difficult from my viewpoint of running the simulations. I guess for Eva, that would make life a bit more painful.
I mean, if you remove potentiation, I think in the Chodix Markram model, it's a 2D equation, so I would say.
Pardon?
It's a very simple model, and the Chodix Markram one.
Yeah. I see what you mean. So right now we haven't done it. For me, it's not a problem to add it. But I guess for my mathematicians colleagues, it would make life a bit harder. No? I don't know.
Maybe we can take one. You had a question, yes?
So with respect to this result of metastability of convergence of the time of extinction towards exponential, I have a question for Christophe, actually. So because I've been working a lot on this on other models, including with Christophe, and I'm wondering how could you, if you wanted to study this kind of thing experimentally in human, for example, where for obvious ethical reasons you cannot put electrodes in people, how could you do? How could you set a behavioral experiment to test this kind of hypothesis?
Well, you could ask humans to be patient enough to do what we ask the monkey, or what we force the monkey to do, and what if this mechanism actually occurs in the brain, you would expect that as you use a longer and longer delay, frequency of mistakes would increase, and this would increase exponentially. So we potentially we could test it with psychological experiments.
And do you think it's doable?
Yes, I mean, let's say yes and no. When you think about it, it's not that complicated to set up. But if I imagine myself sitting in front of a computer screen, staring at a dot, I mean, after 30 seconds, you start thinking of something else, okay? And that would not be the kind of mechanism that plays here to put you out of a fixed point. So we would need a task where the likelihood of mistakes is large enough for short enough time, so that we don't get people so bored that okay they think of something else, and so we need to talk to psychologists for that. But I bet that would be doable.
Okay. Thanks.